Hello and welcome to CEC lecture series on biotechnology. Today I, Dr. Charu Dogra Rawat, Associate Professor at Ramjas College, University of Delhi, would be discussing advanced tools and techniques in which I particularly talk about the polymerase chain reaction. The learning objectives for today's session would be to understand the principle and procedure of a simple polymerase chain reaction and to understand the principle and procedure of various types of PCR methods used in biotechnology. So we had been talking about recombinant DNA technology in which we clone the gene of interest, which is an insert, into a cloning vector for various downstream purposes, for example molecular cloning. So we had been discussing this technology in detail in our previous sessions. For one of the thing to identify this gene of insert, the identification of this insert. So this basically shows the construction of a recombinant DNA molecule. As I said, we have a gene of interest, which is a fragment of DNA. So it can be the gene or a region, but we generally call it as a gene of interest. We have a vector, we clone it into a vector, so we digest it, we ligate it, we clone it, and then we transform it into a bacterial host for molecular cloning purposes. So we have talked this in our previous sessions. To identify this gene of interest, we talked about two methods. The first method is the blotting and hybridization. This again we have covered in our previous sessions in detail. In blotting and hybridization, the genomic DNA is partially digested. It is subjected to agarose gel electrophoresis. The fragments are blotted onto the membranes, the nitrocellulose membranes, and then they are probed for the gene or region of interest. So we have talked in detail about the various steps involved in all these in the entire procedure, and how we can do and blot different biomolecules such as the DNA, RNA or proteins. So particularly when we are talking about recombinant DNA technology, we are talking about genomic DNA or DNA fragment of our interest, but we can also blot RNA or we can also blot proteins onto the membranes and identify whatever is our interest in the particular biomolecule we want to look upon. And we had talked about the details of all these procedures. For the identification of this gene of interest, another method is what we discussed today is called as the polymerase chain reaction. So what happens in this polymerase chain reaction is that the primers are designed for the gene or the region of interest, and then this genomic DNA is subjected to amplification by using these primers. Now I am talking about various terms here. So what are primers? What is amplification? How this genomic DNA is subjected to PCR reaction we discuss in detail. Polymerase chain reaction is also termed as molecular photocopying. So photocopying means producing the copy of itself. So when we use a DNA fragment, then by this polymerase chain reaction, and that is why it is called as a chain reaction, because one after the other a reaction happens and the product is basically multiplied and you get a lot of copies of that target or that template DNA that we want to do. And because we are using molecules such as we use the polymerase and we are using DNA and template, it is termed as the molecular photocopying. So we amplify the small segments of DNA and therefore it is called as PCR amplification. It can be called as gene amplification or simply that we amplify the small segments of DNA. It was developed by the scientist Carrie B. Mullis, who was awarded the Nobel Prize for Chemistry in 1993 for its discovery. This was a very revolutionary method that was discovered. It is actually molecular cloning only. So cloning also involves that we make the copy of a particular gene of interest. But molecular cloning, because it is done at the biological level, everything is basically in vivo. I mean, the recombinant DNA molecule can be created in vitro, but then it is transformed into a bacterial host where it multiplies, and the multiplication of the bacteria causes the gene of interest to also multiply, and therefore we get the copy. So there also it is actually the amplification of the gene of interest, but we use a biological host to do that. And obviously, because we require the bacteria to grow, to multiply, and to reach a particular number so that we can have copies, it is a little time consuming, because it will have to follow the host organism's life cycle, what is the growth curve of host organism. Then it also requires manipulation at the laboratory level, which takes a lot of time to do. When this PCR was discovered, all the concept, the procedure which was involved in PCR was basically created and put in a machine, the PCR machine, and it takes less than 2 hours. Depending obviously upon the number of copies you need, therefore the number of cycles it will run, but it will be like 1 hour, 1.5 hour, the amplification, the desired amplification can be done. So it was a very revolutionary technique that was there, and that is why the Carrie B. Mullis got the Nobel Prize for Chemistry in 1993. Now let's look at the PCR reaction components. Now this PCR reaction is any in vitro reaction that we put in biotechnology. So obviously you need certain components, you mix them together, you incubate at right temperatures, or whatever the principle behind it, you use those steps and you amplify that product. So the main reaction components in this PCR reaction is that you need to have a template DNA. So that template DNA is what we have been talking about as the genomic DNA, or whatever the total DNA. It can be the plasmid DNA, it can be the viral DNA, it can be any DNA which is there. In fact, we will look down in our later in our session that it can be RNA also. So we can amplify from RNA, although we will have an extra step to convert it into a DNA molecule. We will talk in detail subsequently later in the session. But we require that template from which the gene of interest we need to amplify or identify or detect. That is basically the DNA template that we need to have. Then we need to have the primers. Now the primers is generally a set of primers we use called as the forward primer and the reverse primer, and these two primers are basically going to bind to the target DNA or the template DNA, limiting what is our gene of interest. So it is basically at the start of our gene or region of interest, and one will bind at the end of the gene of the interest, and then on the different strands, and then in between it is going to synthesize that DNA fragment which is there. They are called as primers. Because we have been talking in molecular biology also that DNA replication we need priming. So RNA primers go and bind to the DNA and therefore the replication starts. So replication is basically synthesis. Amplification is also synthesis. Replication is generally, there is a one cell, one cycle, there is one molecule that is replicated. Amplification is multiple numbers of synthesis that is occurring of the same strand. But it is also basically the synthesis. So we require the polymerase, we require the priming that needs to be done on the target DNA. So it's no different. It involves basically the synthesis of the DNA only. Then we require what are called as the 4 DNTPs. Obviously, when it is going to synthesize a DNA molecule, it requires the basic bricks which are the DNTPs. So we require these DNTPs which are there, A, G, C and T. We require the buffer and the optimum conditions. So we require certain ions which need to be present. For example, magnesium ions, a particular concentration of magnesium ions aid in the amplification of the PCR process. So we always, for restriction digestion also we saw, for ligation also we saw that we put in buffers. So we give certain ions where the reaction can take place. And we require the DNA polymerase. Now this DNA polymerase and this PCR reaction basically came into being because of the discovery of a peculiar kind of a DNA polymerase which was thermostable. Now what do you mean by thermostable? Thermo means temperature, stable means that it is resistant to degradation. It has got a high shelf life. So thermostable DNA polymerase is the DNA polymerase isolated from for example, one of them is called as the Taq polymerase, which is isolated from Thermus aquaticus, which is isolated from an extreme environment and it can withstand very high temperatures. So we will see when we are putting this reaction, because we are using a double-stranded template, the first thing we need to do is to denature that template. Now you can again recall the basic complementarity or the structure of DNA molecule. You can again go back and look at the sessions describing the temperature, the melting temperature. We talked about in molecular biology about the topology, the melting temperature, etc. We had already discussed that. So the melting temperature, there is a particular temperature at which the two strands will separate, and it is a reversible phenomena. So when you lower down the temperature, they will again complementary bind to each other. So you require a polymerase that can withstand high temperatures, so that when the DNA is melted, then that polymerase is not denatured. So therefore the discovery of this Taq polymerase made this PCR, the reaction, the polymerase chain reaction to come into a reality or to become a technique which we discussed today. So I hope these components are clear to you. Now we will discuss in detail some of the components which are very essential. For example, primers we have been talking about. So primers are basically chemical synthesized oligonucleotides. Now please recall, there is another chemical synthesized oligonucleotides that we use in biotechnology, which we discussed in blotting and hybridization, and it is called as a probe. The major difference between a primer and a probe is that the probe carries a fluorophore or some kind of a detection system which is there, and it basically investigates, it goes and binds specifically to the target. The primers also bind specifically to the target. The only difference is that primers are used to prime the synthesis. However, the probe is only used to go and bind and detect a particular sequence in a pool of sequences or in the entire total DNA, whatever. But it generally carries a detection. We will see that the primers can also carry a detection method, but generally they are termed as probes, and primers are the ones that prime the synthesis of a molecule. So they are chemically synthesized oligonucleotides. They provide the 3' hydroxyl ends by which the DNA polymerase mediated DNA synthesis proteins. So they provide this hydroxyl end. So you know how the phosphodiester bonds are formed and how one base is attached to another base. So when one base is there, it provides that 3' hydroxyl group, the primer, and therefore the synthesis. Polymerases, the DNA polymerases cannot start the synthesis de novo. It requires a 3' end on which another nucleotide can be added. So that 3' hydroxyl group is provided by the primer. The position of the primers along the single-stranded DNA strands defines the total length of the PCR amplification product generated. So I told you that we use actually for a particular reaction, a particular PCR reaction, we use a set of primers. And this set of primers is a forward primer and a reverse primer. So when the double-stranded of DNA melts, then one of the primers binds to one of the strands and the other primers binds to the another strand, and then they synthesize in their directions wherever the 3' hydroxyl group is. So basically, I will show you in the diagram subsequently, but what I am telling here is that these two sets of primers define the length of the product, the amplification that you need. So for example, if you have a genomic DNA and your gene of interest is a particular gene that is only 500 base pairs long, so you need to define that limit of 500 base pairs, so only that 500 base pair is amplified. So if only that is amplified, you will require a primer that will be complementary to that some region, because I said it is an oligonucleotide. The size is somewhere like 18 to 22 nucleotides. So 18 to 22 nucleotides will be a primer. It is going to bind to this 500 nucleotide at one end and another primer at the other end. So it defines that limit which is going to be amplified. So the specificity is provided by the complementarity of the primer to the template DNA, and the length is defined by the forward and reverse primers that you have designed so that they bind at 500 base pairs apart. So the position of the primers, they define the total length of the PCR amplification product that is generated. To amplify a specific DNA sequence, the sequence of the primer annealing sites on the target DNA must be known. Now this is very important, very important, that basically when we are doing PCR amplification, and if you want to identify or amplify your gene of interest, then you should know some sequence of that gene of interest. Now, as I said, for example, we were doing recombinant DNA cloning also, or when we use probing also, then that sequence can be based upon any of the similar sequences which are present in different organisms, different strains, different species of the same organism that you are discussing with. Or even if that the specific is not present, then there might be, it is producing a protein which belongs to a particular family. So you want to look at a dehalogenase gene. So probably you are going to identify some kind of a conserved sequence which is there and try to amplify it. So you should know some sequence that you need to amplify from. So not the entire gene of interest, but whatever, wherever you are designing the primers, because the primers are designed based on the template of DNA. So that template of DNA, you must know some sequence so that the amplification can be done and the PCR primers can be designed. Now the designing of PCR primers involves, usually as I said, the primers are designed in the order of 18 to 22 nucleotides in length. Some of the other parameters which is very important, because primer binding, annealing to the DNA template and extension. So primer PCR depends upon the primer. So the primer you need to design, they are artificially synthesized and then included in the PCR reaction to go and bind to your gene of interest and then amplify it. So these oligonucleotides, the other parameters that needs to be considered involve that whether they have any possible homology to the other regions of DNA within the target DNA. So if you want to amplify a very, very specific sequence, then the primers have to be very, very specific. You should consider that is there a possibility that if you use this primer, then it can bind to any other region apart from your gene of interest. So if there would be any homology, then it is going to bind at multiple places. And you don't want that because you want to identify. So it has to be very specific and you need to consider whether it shows any homology or whether it does not show any homology. The GC content becomes very important because as we talked about in molecular biology, where we discussed about the melting temperature, GC contains 3 bonds, whereas AT contains 2 hydrogen bonds in between them. So it requires lesser energy to break AT bonds than to break GC bonding. Therefore, the melting temperature will depend upon the GC content these primers have, because now they are going to anneal to the DNA template region. Now annealing will depend upon the strength of annealing will depend upon again the GC content as well. Secondary structure, so we talked about, because we use single stranded primers to go and bind to the complementary region of the template, and as we have discussed for the structure of RNA, single stranded molecule tends to form secondary structure because there will be complementary regions within that template. So within that primer, if there are complementary regions, then they are going to form loops. They are going to form hairpin structures, and if they are going to form these secondary structures, they are not going to bind to the template DNA. So that consideration should also be there. Then the presence of the run of various nucleotides, there are polypurine or polypyrimidine stretches are present, then they also should be considered, because again if there are long stretches, then they will be homologous to each other and they can create the secondary structures which are there. Then depending upon whether there are more of polypurines or polypyrimidines, it will also affect the melting temperature of the primers which are there. So these are some of the parameters that when you design the primers, you need to consider it. Now we generally do not design the primers manually, that you do not look at the sequence and try to find out whether there are certain softwares and programs which are present for primer designing. Some of them I mentioned here, such as Primer 3, Primer Quest, OligoPerfect and NCBI Primer Blast, which is again based on the Primer 3 platform. Even Primer Quest is based on that. So these are some of the softwares. So it is generally basically there's a user friendly place where you can just click on that, you can fill in various the template sequence, you can give certain parameters, how much GC content should be there, what should be the range of the, so these are the different things that is basically you include in these platforms, and they give you a set of designed primers which are there, and then you can again further go for quality checking and then give it for the synthesis, give those sequences for the synthesis. So you require the source sequence, you require the range of that primer from where, because you do not need the entire genome, but you require a particular range कि आप सिर्फ इसने from only in this particular region you design the primers from. The PCR product size is given, the primer melting temperature range is given, that we do not want the range to be very high, we do not want it to be 80-degree temperature, it is somewhere around like 50, 55, maximum 56, 58, that is the ideal primer melting temperature. Primer GC content, template mispriming, or self or prior complementarity which reflects the secondary structure, template mispriming which reflects whether there are homology present apart from the gene of interest or not. So all these parameters can be defined by certain numbers and they can be incorporated, and they can be basically feeded in the program, and then it can design a set of primers which can be present. Primer concentration is also very important. A typical PCR containing 1 to 2 picomole of primer and an initial quantity of 10 picogram template DNA should be sufficient to synthesize visible quantities, that is in micrograms, of the amplified DNA upon gel electrophoresis and ethidium bromide staining volume which is there. So, again we have to find out that how much primer should we need. So, 1 to 2 picomole, we should know the concentration of the template DNA that we are using. Accordingly, we will use the amount of primer which we use, and therefore we should have a product. How will we identify that the amplification has occurred? We need to run it. We talked about visualization of molecules. We need to run it on agarose gel electrophoresis, and then if it is visible, so for that so much amount of primer concentration is required. In general, this means that a 0.1 to 0.5 micromolar concentration of primer in a PCR mix is sufficient for successful and accurate PCR amplification in a 50 microliter reaction mix. So you prepare a reaction mix. For example, if you are preparing a volume of 50 microliter reaction mix, you need around 10 picogram of template DNA, 0.1 to 0.5 micromolar concentration of the primer, the two primers, 1x or whatever the buffer concentration which is again provided, and then you mix them, the template DNA, the primers, the buffer, all in a tube which is a PCR tube, and then you add the polymerase which is the enzyme, and then you put it in the machine for amplification to occur. So many cycles of amplification will be there that will be run in this PCR machine which is termed as the thermocycler. So you can see this machine, it has got a plate kind of a thing on the top. This is a hot plate. In the lower inset on the right side you can see a man, whatever these hands are putting in the tubes. You can look at the PCR tubes, so they can be 20 microliter to 50 microliter volumes can be put in a series of tubes, all the reaction mixes, and then they are put in this plate closed from above, and then the program is set where you involve 3 different steps, that is denaturation, annealing and extension, that will cause the product to be amplified. So we discuss these 3 steps in our next session. Thank you.